All right, welcome to TIL Talks with Chris White, where we talk TILs, tumor infiltrating lymphocytes, and all advanced cell and gene therapies, both with patients and caregivers, along with industry leaders. Let's get going. As a reminder, I am not a medical professional or medical expert. I am only a professional and expert on my story and my experience. If you are needing or seeking medical advice or assistance, please reach out to your care team. All right. And so what's up? Welcome back to TIL Talks with Chris White. I have a repeating guest, one of my favorite supporters for TIL Talks, Dr. John Powderly with Carolina Bio-Oncology Institute and Biocytics. So glad that you are back. This is now our fifth recording, and today what I'm super excited about to have you back is we are going to specifically dive deeper into vitiligo, and not just talking about it like we've done before, but we're going to show some slides so you can visualize it. So if you're listening to this, either whether you're on Spotify and or YouTube and or my website, definitely click in on the YouTube spot so you can watch this and see what we're talking about. So, John, I'm going to go ahead and pass it to you. Give a little bit about what we're going to be getting into today. I'm super stoked about this. Let's go.
Yeah. So for the listeners, if you've heard some of these chats with Chris before, I met Chris for the first time at a meeting in near Dallas, Texas, at a Deloitte industry working group for cell therapy. And I chatted with him, and I could tell pretty quickly he had a very strong case of vitiligo. And I kind of surmised that if he was at a cell therapy presentation talk with Deloitte, I said, hey, you must have been a cell therapy recipient of modern immunotherapy that mounted a robust immune response because you've got a lot of vitiligo. And I told him that my life's research in tumor immunology and cell therapy was kind of born out of some interesting experiments I did with vitiligo 20 years ago, 25, almost 30 years ago, with a lot of my mentors. So I promised that this was over about a year and a half ago. I promised I want to show you some slides about why you got vitiligo. Why does it matter? Why is it favorable? Although it's a hassle to get the depigmentation and you're self-conscious about it, it's the best predictor that immunotherapy is working for melanoma. And for your audience, I want to show you kind of a little bit about my research in layman's terms and explain the proof behind the mechanism there. So chime in, Chris, with any good questions. Let me hit share here on the screen.
Yeah, 100%. Like that is the biggest conversation starter out of almost everything that I do is when I meet people and they look at me and they're like, hey, white eyelashes, white hair, but my voice is not matching my look. And then I get into the whole cancer treatment and talk about vitiligo. So this right here, whether you had melanoma or whether you had another disease, but you've done an immunotherapy or you've done a cell therapy or anything where it would cause an immune response to where you would get a vitiligo, which takes out your pigment. This right here is going to give you a visual on both why and how it happens and the positive response behind it. So, John, take it away.
Yeah, thanks. So everything here is in the public domain. I've presented a lot of slides over many decades about immunotherapy and vitiligo. And this was the last scientific talk I gave to young students. This was at Georgia Tech. They had a industry symposium for immunoengineering last year, and I was one of their presenters to their PhD students who were growing immune cells for different immunotherapies. So basically, it's a little background about how I was inspired into cancer immunotherapy. We'll talk about that. Why these animal models of melanoma and immunotherapy, why vitiligo is one of the best teachable basics around melanoma immunotherapy, and then why I founded a research clinic here in Huntersville, and then a little bit about some of the phase 1 trials we have done when we first started the clinic. We may get into that second half after 2005 at another talk, which is great. That's more modern immunotherapy. But this first half of the talk will really be about the animal models and what I did as a young med student and resident and fellow understanding immunotherapy and why vitiligo matters. So for me, my story started when I was a senior at Georgetown Med School, working for Steve Rosenberg in his research lab there
Legend.
at National Cancer Institute in Bethesda, the NIA. And it was my dream to go work for him as a rotation to learn about how he was growing immune cells, how he was developing immunotherapy. And he is that's considered the grandfather of modern immunotherapy, who's still alive today, and his work ethic is unbelievable. He's 83 years old and had been working in a lab pretty much nonstop. He
Still
was the
going.
yeah, the youngest MD PhD director ever of the surgery branch, the National Cancer Institute up there, and is just everyone that trained up under him is like the diaspora of all these research labs and biotech companies. So just an honor to even talk about what I learned from him. But this will resonate with your viewer. So my first week at the NCI, he asked me to go down to the basement of the National Cancer Institute and read a historical book in what we used to look up the Index Medicus. You'd look on all the indexes every year of everything published. This was before you had internet and,
Yeah.
PubMed. And he said, there is this book from 1965. It's in the basement. Go down the library and you find it. And then tell me what you learned. And I went down. I said, what kind of exercise is this? And it was remarkable. It was a historical chronology of hundreds and hundreds of cases of patients who from 1900 to 1960 cured themselves of cancer, like literally cured themselves. This is before we had modern chemotherapy, before we had great modern surgery, or certainly before we had modern immunotherapy. And it was published by two surgeons, Everson and Cole, and they described well-documented 176 cases over those 60 years of clear, convincing evidence of patients that had cured themselves of metastatic stage 4 cancer. And it turned out that the incidence of this autologous, automated self-cure was about 1 in 60,000 to 1 in 100,000 cancer cases. So it was very rare to cure yourself. And these cases, remarkably, in many of them that did get cured, it wasn't that they just got cured out of the blue. It wasn't like they went to a prayer session or just, drank some elixir. What had happened that seemed to precipitate the cure was an immune catastrophe. And the immune catastrophe would be something that during those 60 years would be akin to like a surgery went wrong, like a bowel perforated and led to a massive infection while they were trying to resect a tumor. And the patient almost died from the infection and the perforation, but somehow recovered from the infection. Antibiotics were around at this time. They recovered from the infection, the wound healed. And then months later, the tumor began to regress and disappear. And then the patient never died of their cancer. Although they were in the abdomen, they saw the cancer, they resected part of it, they left part of it behind, or they couldn't resect it all. So it seemed like the pattern of all of these 176 cases were associated with some immune event, a catastrophic event. A perforated bowel was one example. Another example would be a patient with lung cancer would have a terrible case of tuberculosis and had high fevers for weeks and months and almost died from tuberculosis. But then tuberculosis recovered and their cancer regressed and disappeared. There was other cases where patients were given a mismatched blood transfusion. This was right. Blood transfusions were starting to be developed after World War II, but they didn't always have the match exactly right. So some patients after surgery would need a blood transfusion and mismatch, terrible reaction. And the patient almost died from the transfusion reaction, high fevers, rigorous chills, hemolysis.
Would that be the graft versus host?
Graft versus host. Yep.
Yeah.
They probably couldn't weed out the white cells. So it was the donor cells maybe were contributing. And then there were other examples where some women who had gotten usually it's breast cancer at the same time that they're pregnant. That seems to feed breast cancer sometimes. But then after the child was delivered, there were complications and infections with the placenta and a very hard childbirth. And the mother almost died from the childbirth, but had complications and infections. And then lo and behold, the cases of cancer that I remember were like a breast cancer have regressed. So a lot of these events were very clear immunological events. And in many of them, very many of them, especially in the melanoma cases, patients would get whitening or vitiligo of their whole body as the tumors regressed. So that if they mount an immune response, if they mount against the pigment cells, right, the pigmented melanoma cells, they would also cross-react in, as a bystander, collateral damage. It would kill the melanocytes, the healthy normal melanocytes, which are the pigment cells that give us pigment. So that was an inspiring book to read and to report back to Dr. Rosenberg. And I remember asking him, I said, Dr. Rosenberg, why does this matter? What is it? Why do we focus so much on melanoma in vitiligo? Why does that matter? Why is most of your research on melanoma? And he says, quite simply because we can see it, meaning you can see the immune system attacking the
You have a
melanoma
visual.
inflamed in the skin. It might look red, hot, swollen. And then the patients would start to get vitiligo in splotches or circles around their melanoma. Or in other cases, they could even touch and surgically remove the melanoma so they could examine the immune infiltrate. So this was why in the early days, although, Everson and Cole surgical, those surgery books, any cancer could potentially cure itself if there was an immune catastrophe. Melanoma was ideal as a model because you could see it happen. And oftentimes the tumor itself got infected. And then they could inject more things into the tumor to make it become more infected and stimulate an immune response. And some of these old adjuvants you see there where they would be mycobacterium, which is kind of like tuberculosis, heat killed, but it still was dead bacteria or live bacteria right into the tumors that would cause inflammation and little mini infections. And that would stimulate topical tumors, little areas of tumors regress, sometimes throughout the whole body as well. So I thought that was inspiring. And then that's why surgeons got involved with resecting the tumors, seeing how much immune cells were in them, and then learning the laboratory tricks around how to grow those immune cells right out of the tumor, how they would kind of homogenize or dissect or digest the tumor, let the immune cells grow out, and then plate the tumors on a different plate, pour off the immune cells into a different petri dish, and then study them, and then mount that immune response all over again. Say, hey, can I add these immune cells back a few weeks later to those tumors that were growing? And you could recapitulate the immune system in a petri dish. And so those early days led to the discovery of tumor infiltrating lymphocytes, which TIL, hence your name of your podcast.
It's not just today I learned, it's tumor infiltrating lymphocytes,
Yeah.
everybody.
So if you learn one thing, this is how they were discovered by these surgeons. Surgeons really led the modern immunotherapy push, and most of them were the early learners in the surgical branch at the NCI. That also led to the discovery of the hormones that made the immune cells grow, interleukin. Right? Leukin is white cell, inter is like communication. And they gave these in numbers. You were communicating white cells to stimulate each other. Interleukin, we call that a cytokine. Cyto means cell, kine means like a hormone or a peptide, a protein that is informing another cell to do something. So interleukin is of the class of families cytokine. So you'll hear those
Cytokine
words a lot.
release.
And cytokine release or lymphokine would be like a protein that you could stimulate immune cells with, lymphokine activated killer cells, which looking back, interleukin was the lymphokine, the old name for lymphokine, and that it would stimulate T cells. Or at the time, they thought they were natural killer T cells,
NK cells
NK cells, or
nowadays, right?
T cells. So this was the early days that they were doing this in the late 80s, early 90s. And they were able to grow up those cells and give them back to patients. And remarkably, they saw them work in 6, 7, 8, 9, sometimes 10% of patients, quite remarkably, and study the patterns. And so when I trained under Steve Rosenberg at the NCI, and I had my other mentors there at the time, Patrick who's now runs the Moffitt Cancer Center, and
Yes, sir.
Nick Restifo, and a lot of other great leaders there that I got to learn from them. One of my projects then in 1994 and 95 was to take pictures of the cancer patients with melanoma that had gotten interleukin 2 or tumor infiltrating lymphocytes or lymphocyte activating killer cells or vaccines like those MAGE and MART melanoma vaccines. And when they got regressions and got vitiligo, I would take them down to the photography center in the bottom of the National Cancer Institute. We have a black light, like when you go to Halloween parties, and everything lights up. So they had a black light to help them kind of do different wavelengths of different black lights, and had photographers take pictures. And we would capture the patterns of the vitiligo to see if there was any clue about, does the vitiligo pattern matter as it relates to where the primary melanoma was versus where it might have spread to. So this is a great project for a med student back then, and it inspired me. And Dr. Rosenberg would call this breaking self-tolerance, because the immune system has to not only fight the cancer, but to really work, it's got to break through that self-tolerance and kill something kind of normal, like kill part of your normal self, like the melanocytes. So that was an early concept that to have it work, you had to get vitiligo, meaning break your own tolerance Like you getting vitiligo, Chris, you broke tolerance. You broke your self tolerance because you could not tolerate your own melanocytes. Your immune system overcame them along with killing the melanoma.
Just not trying to get off a topic, but real quick, we just want to ask because my primary source of my melanoma was nowhere near where the first signs of my vitiligo was. And so I'm kind of curious because I had anal rectal mucosal melanoma, but my first signs of vitiligo was around my face, around my goatee area, I guess, and then a couple of spots on my hand. I didn't notice it around other areas of my body. So were y'all able to determine at that time through any of those studies that that was vitiligo was correlated with the source of where the initial melanoma originated from, or
So great question. So that was exactly my hypothesis of, well, of course, the presence of vitiligo is like the strongest favorable prognostic factor. I wanted to take it to the next level and say, what about the pattern? Does the pattern matter? And I still think the pattern matters, but we haven't gotten enough pictures. The experiment kind of winded down after I left.
you got to graph it, right?
They had some 50 pictures, and everybody else has validated that, yeah, presence of vitiligo matters. What did go, and we have some slides about this, is when you look at animal models, yes, the pattern even matters more in the animal models. In some of the animal models, guess what? They get anogenital melanoma. You'll see their pattern. It's coming up. At least we'll see what the animals told us about your question. So yes, I think that to cut to the chase, anal genital or mucosal melanoma that's in the mouth and the genital, those are such stemness with a very primordial stemness melanoma antigens from the earliest beginning of where the melanocytes started. So that antigen that's in those melanocytes is probably in all melanocytes. Hence, that's why you got full body vitiligo,
Yeah, it
including
actually
eyelash.
went everywhere.
And you'll
Okay.
see in the other patients where they did not have anogenital mucosal melanoma, their antigen repertoire of their vitiligo pattern was more focused on certain dermatomes.
Well, I'm looking at your slide right here with this picture, and it looks like it's forming directly around a lot of the actual the moles or
Yeah.
the melanomas, which would be like for cutaneous patients, which is the
Yeah.
majority of melanomas,
So a lot of
would make
melanomas
sense.
start as a dysplastic node. So the picture there is classically a dysplastic nevi. This is like a pre-melanoma.
Okay.
But some people that get a pre-melanoma, they probably have a little teeny baby melanoma in the dysplastic nevi, and it's getting just mutated enough that the immune system here in this picture, the immune system is already mounting an attack before it has a chance to grow into a full-blown full melanoma, and it's already starting to kill off the dysplastic nevi. There are other cases in other pictures where somebody would get a melanoma on their right shoulder, it would regress, and then they would start getting halo nevi of all their nevi. Like the nevi would be, because they're so dense with melanocytes that were probably a little bit dysplastic, and those would start getting a halo nevi around it. But that picture of the halo is really good because that's an immune attack of the dysplastic nevi, the pre-melanoma. And then about a couple of weeks later, the pigment's gone. That
Yeah, it just disappears like it wasn't there, right?
Yep, exactly. So let's take you to the next slide so I can show you a little bit. So these were some of the examples of the patterns of patients with melanoma and the publications that were around this time. The point being is that normal tissue antigens, right? Not just the tumor antigens, right? Which are the mutated antigens that are specific to that tumor, but normal tissue antigens can also be the target of a robust immune response. And in fact, if you can break self-tolerance and kill a little bit of yourself, that's a really good thing. Now, we don't want anybody's immune system to kill themselves.
No, you build it down to break it back up, right?
If you can just break just enough self tolerance, what we tell patients, you get a little more than just a fever, flare, flu symptom, fatigue. You get a little bit more of a very mild, teeny amount of lupus, a teeny amount of an itchy skin rash, a little bit of vitiligo. And even in the blood work, like you could even see a little bit of some borderline lupus. Sometimes those are favorable autoimmune tendencies. Like we don't want to wish full blown autoimmunity on anybody, but a little bit of autoimmune tendency in a immunotherapy patient is very favorable for a good response. So, and the reason there are patterns in the first place is because this is even more interesting, is during embryology, when we're developing the womb and as an embryo, the nervous system is among the first things to develop. We learned if you ever took immunology class, like The embryo kind of develops, and then the eyes and the brain are very soon thereafter. And the first thing that gets pigmented is the pigment in the eyes, right? And the eyes is part of the brain. And then the brain, as it sends nerves down the neural crest or the spinal cord to these little budding limbs, every dermatome, and everybody knows what a dermatome is, right? That's like the stripe of the nerves that go down each layer of your vertebrae. So each so they do now. So each dermatome, which is a different vertebrae, each vertebrae has its own nerves, right? So and each one of those vertebral nerves that grow down come from the embryonic neural crest. And as the embryo is developing, all through evolution, all mammalian animals through evolution, each of those dermatomes in each vertebrae is slightly different. It has a slightly different antigen repertoire, which is why animals get stripes and animals get pigment patterns. The same
Because it
reason.
all stems from their vertebrae, and then it eventually gets to the exterior, and that's why you see it on the outside, and that's why you see the stripes.
Bingo. The same reason a calico cat, like the spots are kind of
The
blotched.
most prominent wherever their spine is on their body, and then
There's
it spreads out
zebras,
from there, correct?
yeah, or piebald animals. You have piebald cat, a piebald horse, piebald dog, and you see they have the white mane right here, the white feet. All those are because the way the neural crest migrates down to innervate those, they have their own clone or colony of pigmented melanocytes. Hence, their antigen repertoire, their
Which
antigen surface are different.
and this is why on this second picture, why you can see under the black light, why the vitiligo is, populating most prominent here, like, closest towards the topper end of the spine, correct? And then that's where it
Different
first
dermatomes,
show
like,
up?
some patients would get a melanoma that started on that side of their trunk, and then you would see their vitiligo pattern would be on that side of the trunk. Like, it was very
Uh,
specific to
okay,
the clone of the melanocytes
makes
that
sense.
went to that side. So even there's a hemisphere of your body, their tumor antigens or their melanocyte antigens can be slightly different. And the hypothesis is that the tumor that arose from that dermatome, the melanocyte that gave birth, that developed that melanoma, when you mount an immune response, the immune response should overcome not only that melanoma's antigens, but that dermatome's melanocyte antigens. Hence the dermatomal patterns. And like some of the gentlemen, it might be the trigeminal nerves, or it might be like that gentleman there where you can see it's kind of like a V. That's a dermatome actually right there. And as it goes down his chin there, that's another dermatome. So those are different melanocyte surface antigens that the immune system can recognize to attack or not because they're different. And so it's almost giving us a window into what's the immune system doing as it attacks the melanoma based on different antigens.
Kinda be a predictor of what's happening, where it's could be possibly going.
So it was very interesting. So when you look in the literature, and this is part of my residency. That was in med school with Steve Rosenberg and taking those pictures. And during my research, trying to learn about this, I found out there are other animal models that cure themselves of cancer. And I hadn't, we didn't learn that really in the rest of med school. It was only about learning about that studying vitiligo with Steve Rosenberg. Is like, hey, some animals can cure themselves of cancer. We always learned elephants never get cancer, right? Because they have all these copies of TP53 and there's reasons why
Yeah.
it's get cancer. We kind of learn, oh, bats never get cancer because their immune systems are different. Like these are other things. And you start hearing some other animals like rarely get cancer. But it's funny, there are some animals that routinely get cancer if they're inbred, and those animals can actually cure themselves of cancer. Really cool. So I was like, and I learned not only do they cure themselves of cancer, but they get vitiligo.
Yeah, look at that. What is this, the bottom right corner picture? Like,
Yeah.
that's stems like, that's the hair color, but that comes from growing out of the skin. Like, if you see me in person, I got the salt and pepper going on,
Yeah.
or if you've seen my checkered skin, but those spots where it's dark hair coming out, but the other spots, that's the white from the vitiligo, and that is super unique to see this. Is that a cat or what?
So no, this is
Is that
a
is
swan.
that a pig?
So porcine pig, swan.
Okay.
So this was so cool when I found out about it before I left Georgetown and Steve Rosenberg. I says, Dr. Rosenberg, heard about this herd of swine that are inbred and they routinely get melanoma and cure themselves. There's only two herds in the whole country, probably in the whole world, that the herds are maintained. One was at Missouri vet school, and the other was at Texas A&M vet school, like large animal vet school. So I said, oh my God, I'm doing my residency in Houston. I'm going to, on my weekends, and talk to the veterinarian. I'm going to learn more about these swine. And I'm going to, whatever I learn, I'm going to tell Dr. Rosenberg to kind of keep this project going in the back of my mind because there was clues here. To me, this was like a clue on how can humans cure their own cancer. Let's look at what the pig does.
Yeah.
How can we do that? Because I was seeing the humans get vitiligo. I was taking their pictures and we were seeing the positive responses. I'm like, there's a major clue going on here. What can we learn more? So this is
Uh,
swine. It's called the Sinclair swine. It's not the kind of swine that it's not like an oink oink pig that you eat. It's kind of like a
I was
wild
gonna say
boar.
it's one of the small one is in the
It's
second
not
upper
as
right
small as a
picture.
guinea pig, and it's not as crazy looking as a wild boar, but it's
Okay.
certainly not like a regular domestic oink oink pig that we eat. But as the melanoma model, it was awesome. So the first month of life, when this pig is born, it develops cutaneous melanomas at multiple places in its body. It's almost like it's programmed for its melanocytes to turn into melanoma. Why? We don't know that, although people are looking into it. We don't know why it gets
Because
melanoma.
they have a
They
lot of hair. It's not like their
don't.
skin's exposed to the UV rays. They have
Well,
melanocytes.
there's probably a gene driver there, which that's another story. That's a
Yep.
genomics, but
We could do another episode on that.
yeah, but from an immune response story, this is beautiful. So during the second month of life, the pictures that they're holding, that guy in the upper right hand, the upper left is the nevus, like the mole, the melanoma really just barely started. Then a couple months later, when the pig's like 2 months old, it grows into this exophytic, like a cauliflower. It grows as a melanoma protruding from the body. And then by the fourth month of life, between the second and fourth month of life, macrophages, which is a type of immune cell, it's an old, let's say myeloid immune cell. It's not the modern lymphocytes. It's an old myeloid lymphocyte, an old myeloid immune cell, macrophages. It'll invade the melanoma and start to get it inflamed and begin a little bit of regression. And then by the fourth month, some of those melanomas start to grow again or they grow in other places. So it's almost like there's a partial regression with the myeloid infiltrate. It's like an almost kill, but not enough. And then the pig develops widespread metastatic melanoma that are about to threaten its life. And then a second infiltration of a different kind of immune cell comes into the rescue. That's the lymphocytes. So this is where the teaching point is. It's like you got to have a robust myeloid infiltration of macrophages, monocytes, dendritic cells, natural killer cells first. And then you might get a big flare, ulcer, inflammation, and it still may metastasize. But then the lymphocytes come in, which are the adaptive immune response. They come in and they get educated. And then they can clonally expand and mount a bigger immune response. So while the tumors are metastasizing, then the real army comes in, the bigger army of the lymphoids, and then that's when the vitiligo starts. First at the primary site, right in the dermatome, and then it spreads to all the other metastatic sites. So it gets more interesting. So if you go to the next period of time, if you follow these swine another couple years, just to prove to people that they are indeed metastatic, we would sacrifice the pigs, or Dr. Amos would and his team, and I was privileged to watch and get the pictures. Those are little pigmented melanomas that had spread to all the other organs in the upper left, lung, liver, just to show some of the pigs, they were clearly, convincingly metastatic stage 4 melanoma, right? During those next 2 years, as the lymphocytes expanded and clonally proliferated, they first killed the melanoma in the primary dermatome, consistent with my hypothesis from that. And then they started killing the rest of the dermatome, right? So
So
they would
this would be
literally go
like
out
hitting the trunk of the tree and then branching out to the
and then
other
branching up
branches
to
and
that whole
leaves,
dermatome.
right? So
So
you
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 can
start
see
with the
here, so
trunk.
this up in the upper right, this pig's primary melanoma was right there on his cheek. But first he got a good white spot there, but then where was part of that dermatome, the trigeminal nerve, like that patient in the picture you saw the trigeminal nerve, that's his next spot of vitiligo. So you could see it's that whole stripe would get the vitiligo in that dermatome, consistent with the specificity of the antigen in that dermatome. Then down here on the lower right, this was a different pig, another pig that got melanoma here. He had regressed that melanoma, immune system successfully attacked it and won, but then look at where he started getting his vitiligo pattern in the
Yeah.
exact
Spot on the
contralateral
opposite side.
dermatome, showing again that the specificity, the very unique antigens to that dermatome matter for a robust immune response. And then in the lower left down here, this is what happens when the pigs live long enough is that their immune systems keep stronger and stronger, and they basically get vitiligo everywhere. And then this is sad, but this is how strong their immune system is. Ultimately, after like 2 to 3 years, even the pigment cells in their eyes get attacked and they lose the pigment in their eyes and they go
I
blind.
was gonna say, is that essentially like cataracts, which would be caused, like, in a human case,
Well,
a uveitis? Because
we're,
immunotherapy does
yeah,
cause uveitis, which does lead to cataracts.
worse than cataracts you can remove. And, but this is the back of, yeah, it's the retina in the back, the pigmented cells in the back of the eye, they get attacked. And so this would be like an immune system so strong that yes, it cured itself, the host cured itself, but it got wiped out. And I bet if we could talk to the pig, he probably got deaf also. Because there have been some human experiments. There is some They can tell you. There's some pigmented cells in the inner ear that are part of the evolutionary mammalian nervous system that they might have gotten attacked as well. There's been rare cases in humans when, in the early days of immunotherapy, it matched such a robust immune response. Vitiligo, melanoma regression, everybody's celebrating. And then you get retinitis and even some deafness. And luckily, nowadays we have medicines to reverse that quickly. We now know better. Like if we knew, you know, 20 years ago, we'd say, give the poor pig steroids. Let's prevent him from going blind or deaf. We would try to rescue his own immune response if we could. We just didn't know that. That was the natural history of the pigs who have taught us a lot, right? So thank you, pigs.
Thank you.
So next gets a little bit interesting. So guess what?
These are horses.
There are more animal models that cure themselves of
Born
metastatic melanoma. Unbelievable. So in me, this was so amazing because
It is,
after I did
dude.
my residency in Houston in MD Anderson, I spent all my extra year there, learned immunotherapy at MD Anderson while I was studying the pig. Then I did my fellowship at Chapel Hill. So Chapel Hill was
Okay.
right near horse country. It's horse
Yep.
country. And there
Yeah.
was this old-fashioned veterinarian who knew a lot about horses. And I read in a veterinary journal, just doing a literature search, veterinary journal about a horse melanoma vaccine. And I'm like, what the hell's going on there? What the hell's going on? And he was given horse melanoma. He was actually working with another doctor in Florida, which is also horse country, North
Oh, yeah.
Carolina and Florida, big horse
Oh,
country,
yeah.
and South Carolina too, I guess, and other places.
It's like,
And there is a horse that spontaneously gets anal genital melanoma routinely. It's the gray horse. These gray horses, unfortunately, they get melanoma, but they start to regress their own melanoma with the same type of macrophage myeloid infiltration. Then it metastasizes up and down the aortic lymph nodes. So if you do an anal exam, I didn't do that, but the vets would tell me about it. They could feel the melanomas up and down the aortic chain, and sometimes it would cause anal blockage. But during this whole while, they could see that the vitiligo was happening more diffuse. And you could see that it would start a little bit speckled, and then the animal would eventually become a white horse.
I was about to ask because I was just looking at this. I'm like, this is a complete black horse, a little bit of vitiligo around the face and everything. You're starting to see it over around the leg. But then if you look at this bottom middle picture,
Yeah.
that is what I would consider a complete white horse. Yes, a little bit of black around the legs and the tail, maybe up near the mane. But basically, I would consider that a white horse. But this is the same horse,
They're called gray horse. This was, well, the two on the right
and that's the
are
vitiligo
when
from
they
it, though,
are
right?
like gray, and they're getting, this was when on the right is when they were getting the anogenital melanoma that had started to spread to the hind leg, or it would metastasized from the anus down to the
So
hind
this
leg.
was a sign of the
This
immune
was
response. Okay.
on the lower left was a picture of one of Dr. McCashin's other horses that he treated that was 5 years old, or that had gotten fully vitiligo white, but had survived its anal genital melanoma. And he was involved in some of those melanoma horse vaccines, which is now becoming standard of care. And you won't believe this, Chris, but there is now an official FDA approved melanoma dog vaccine for dogs that get melanoma.
I've heard.
And
A
Yeah, and
little
they
bit
get a
about
little bit
that.
of vitiligo as well, and it's very powerful, very good melanoma vaccine.
Coastal melanoma is actually quite prominent in dogs, in canines. So
And
there's
so
a
the
lot of
FDA
studies within that.
specifications,
Yeah.
manufacturing, regulatory trials, all that. So it's just amazing how these animal models they're teaching us. And so the next animal model, this is more of a laboratory created animal model. Okay.
All right.
So this is the mice animal model that we routinely did in our lab at Chapel Hill when I was there, where you can't wait for a pig to grow up and regenerate a pig and have progeny. And you're waiting like 2 or 3 years to like study that. In horse, you got to wait 5 to 10 years to follow the whole. So
Yeah.
this study melanoma in basically 3 to 6 months.
Is this
So
set of slides from the late 90s or early 2000s, right?
this was during the 19, I would say late 80s, 90s. There was discovery. There was kind of like a random discovery that one random mouse got mouse melanoma, and poor mouse. But a smart scientist in some lab said, you know what? And I can inbreed that mouse so that it has very close cousins and then inbreed the cousins. And basically, you get what's almost identical twins. If you inbreed them enough, you get like a whole army of identical twins mice that are so similar that you can then transplant a tumor, a mouse tumor, into all the identical or syngeneic mice, and you can do experiments on them. So the one poor mouse that got the first melanoma, um, they cultured his melanoma in a petri dish and it kept growing. But then that mouse that got the melanoma was able to be inbred. And so they now had an army. Years later, you can buy this type of mouse.
Were they able to clone it, I guess? Or
Yeah, good question. It's so identical, it pretty much is cloned. We have done at Chapel Hill, we put in genes. We replicated, yeah, they were in a mouse cage and they just
okay.
repopulate every other month. They have babies, and we could put in genes into some of the zygotes, if you will, or we would hire somebody to do that. Different color genes like red fluorescent and green fluorescent proteins, and you could then put into the mice to make them fluoresce different colors. And then you could transplant tumors and transplant immune cells between the mice that were all identical siblings or identical cousins, and trace, where do the immune cells go? Where does the tumor go? Does the immune system attack it? And if you inject the mice tumors into the mouse tail vein, it would go to the lung metastasis, right? And
Yeah.
and that's on the right lower is the lung metastasis,
I was
which you
gonna
could
say that
trauma.
that Mets looks gnarly. Like
Yeah,
all the
the
dark
mouse.
spots are the metastasis, right? Like,
Yeah, and this was the mouse could, they you have to sacrifice him before they suffer. So the mouse was sacrificed, but he had a lot of tumor in his lung. And
yeah,
over on the left is the lower left. That is what melanoma looks like in a petri dish. And melanoma is
yeah.
probably the easiest thing in a petri dish to under a microscope to tell it's melanoma because guess what? It has its own pigment. So there in the center, it's got the brown melanin pigment. So we know we're growing melanoma in the petri dish. We could then inject
You get a
it
visual.
in the mouse tail
Yeah.
vein. If we wanted to inject it in the hind leg, not the tail vein, but in the hind leg, it would grow into a tumor nodule, and we would measure it. And then if we wanted to grow immune cells, we would actually make green immune cells with the green fluorescent protein, different color. You could inject those into the foot pad to see if they migrate to the tumor, or inject them right into the tumor to see how they behaved. And so those were a lot of the experiments we did at Chapel Hill. And then, so this was the animal model, but here's the cool thing that it taught us is if you break self-tolerance as a mouse model, you, the mouse, cure your own cancer, or we laboratorily cure it with our experiments. You also break self-tolerance. And there might be a little clues that it's vitiligo, dermatomal, but you can see all of these animal models going back to the 1990s. This was like the excitement around modern immunotherapy during the 90s. These are all different labs using the same mouse model. They all were comparing notes. So my mentor at Chapel Hill, John Soroti, he was doing these, fluorescent
Horowitz.
mice and
I mean,
knocking out genes and adding in genes. And we did some of the dendrodixol work. So we even did it. But here's what's interesting. All those mouse models were kind of cool because they did break self-tolerance. Yes, they get vitiligo, but most of them were stepping on the gas pedal to stimulate the immune system. So this is the teaching point.
step on the gas,
On
release
the
the
gas
brakes.
pedal,
It's
give
all
more,
yeah.
yeah, more gas pedal. You could stimulate the hell out of an immune system, those mice would get a mouse fever. They would get mouse flu symptoms. I'm sure they didn't want to eat. They were sick because we could step on the gas pedal with more vaccination, more cytokines, like we even did in the human, step on the gas pedal with more cytokines. And it worked maybe 10, 11, 12% of the time. But something changed in 1999, which is instead of stepping on the gas pedal with the mouse models, we figured out the brake of the immune system, released the brake.
Is this the bottom left
This is the famous,
picture
yeah,
on the
James
slide
Allison.
that
This
we're
was the famous experiment.
yeah.
James Allison, he was the first to figure out what is the brake of the immune system. So before this, is modern immunotherapy, gas pedal, every year we were discovering a new gas pedal. Gas pedals were on the front page of Time magazine. Interferon one year, we're going to cure cancer. Interferon another year. Interleukin, we're going to cure cancer. Another year, Steve Rosenberg wrote a great book, Transformed Cell. We're going to cure cancer with gas pedals. It did a little bit, and it was a big step, a big 10% it worked. But it wasn't until we learned what was the brake of the immune system that was holding it all back. So once we could release the brakes, now you could break self-tolerance. You could overcome self-tolerance and destroy whatever the hell you want.
That's why I want to highlight to everybody as you're watching this is bottom left picture where it has the green lettering and it
Yeah,
says block. All
exactly.
right. Please pay attention to that
Because
because that is the
yeah,
turning point of what we're
now the
talking
immune
about here.
system is unleashed when you release the brakes and you have a gas pedal. Think about that. You got a gas pedal and release the brakes.
Yeah. You
That
put on the
car,
gas, but then you just like release the brakes a little bit. That means that your control.
bingo. You got full control. The only thing is you got to be careful. You better wear your hat. You wear your helmet.
Yeah. You don't
You
want to be
better make
slamming
sure
into the
you better
sidewall, you
make
know.
sure you're not running into your organs.
No. You gotta be. It's very controlled, but
But
this is a
that's
more
how
controlled way to
powerful,
do it. Because you keep your foot on
powerful.
the gas and release the brakes as you need to, so
Yeah.
you can let a little in, let a little out, like and control it.
And as an example as to how powerful that CTLA4 is, we just have an antibody and block it. But if you take a mouse and you knock out CTLA4, so it has no brakes, that mouse is born with lymphoproliferative, like lymphocytes are growing like crazy, lymph nodes, and by a few weeks of life, it dies from fulminant autoimmune catastrophe with lymphocytes proliferating everywhere. It's bad. That's how powerful, like just as if you take the gene away from the mouse. I mean, it'll still be, it'll go through embryology, it'll be born, but it just has a horrible first life. So you can't block,
Yeah,
you can't knock the brake away. That brake is so important. In fact, in humans, if you give CTLA4 as an antibody, and this is why there's major black boxes, it would
you
make a
have to.
fetus spontaneously abort because there'd be no brakes and the mother's immune system would attack
It
a
would just
fetus, catastrophic immune attack. So it just, it's in here to show it worked. It was the beginning of the next phase of modern immunotherapy. It explains why you got vitiligo, why you're probably
It's a
alive.
great timeline right here, just to give a little breakdown because this didn't happen overnight. It's been years and years and years
A
and
long,
progress. Yes,
all these leaders in they still go to all the annual Society of Immunotherapy meetings. Everybody is in the know. It's a great community. And Dr. James Allison, who won the Nobel Prize, I think 2000, and he plays the harmonica in a band called the Czech, which is the class. So he's a great leader in the community, and it was a privilege to work with. And our clinic was among the first to give the CTLA4 drug. And we were on the phone calls when we had our patients responding. And so Steve Rosenberg's clinic did the early studies, and we were the next clinic to open, the first community physicians clinic. And we were part of those early calls. And that was a joy early in my career to do that. So the story gets better. This shows what CTLA4 is that releases the brakes, just for teaching.
activated T
So
cell.
these are two cells. I would say this is the synapse of the immune system, if you will. Synapse,
The synapse.
neural synapse is how they communicate. This is how the immune system communicates with itself, or we say how the immune system gets educated. Okay. So the blue cell is a lymphocyte. That's easy.
Okay.
Lymphocytes.
Yeah.
The green cell is a dendritic cell, which is like a macrophage monocyte. It's a cell that is presenting antigens, like tumor antigens that it picks up like in the tumor and brings to the lymph node. This is what goes on in the lymph node. The dendritic cell, the green dendritic cell, brings back tumor antigens. The middleman cell or the professional educator, they present antigen. They're like bridge the modern lymphoid or adaptive immune system, which is, mammalian biology, like after the sharks forward. And then they bridge the old pre-shark myeloid immune system.
Gap bridgers.
Yeah, the gap bridge. So, and these cells are very important. But anyways, this is a dendritic cell. When a dendritic cell educates a naive lymphocyte on what to attack, it has to teach them what to attack. So the dendritic cell hugs the lymphocyte. So it's giving it a hug, and that red there is like the abort signal, the brakes. And that abort signal is so important that when the immune system is getting educated on what to attack, 99.99% of what is educated in the immune system usually ends up with a abort, don't attack it, because it's mostly self antigens, right? Right. So
It's like the
God made
battle
us with
plan attack, you know what I'm saying?
great.
Like,
God made us with all these, balances, right? Like, so if we step on a thorn, we can attack it, it's foreign. But if you get a little inflammation, don't attack it, that's self. So we evolved to have these very elaborate breaks, and CTLA4 is the most important one. And it should be, because if you release it, God forbid, you get immune catastrophe too much, or in the mice, when you knock it out, it's just bad. So we release it in bits and pieces when we give the checkpoint blockade. But this is that synapse of antigen presentation happening. And the red is the CTLA4 itself, giving the signal, abort, 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 put on the brakes, don't kill me, I'm too important. So this is kind of an overview of the gas pedal and the brakes in a very simple way for your audience. Like CTLA4 and PD1 are the biggest brakes in the immune system. In the lymph node, the brakes, called central tolerance, the brakes in the lymph node is the CTLA4, which stands for cytotoxic T lymphocyte antigen 4. We didn't know what it did when it got its name, so it just got that name. The
right.
PD1 got its
So,
name after we learned what it did, programmed death. Programmed death means it would be involved in the lymphocyte killing. Programmed death, like a deadly handshake of an immune cell killing. It was involved in that pathway of why do healthy cells don't get killed by immune cells. Like, so the PD was the brakes of our immune system outside the lymph node. So PD1 and PDL1 kind of keep our body in check in the periphery. And in the lymph nodes, when we're draining antigens through dendritic cells to do presentation in the lymph node, CTLA4 keeps us in check with the brakes in the lymph node.
because sometimes you need to speed it up and sometimes you need to slow it down. But at the
Yep.
same time, you got to think about it like you're in traffic. All right. You're trying to weave in and out of it. You need to speed up, you need to slow down, but you got to keep moving and you don't want to come to a dead stop. But you got to find that middle ground to where you can keep progressing. And this is essentially what it is. You got your CTLA4, you got your PD1. Patients that have done combination immunotherapy that are listening to this, it's essentially like trying to figure out the balance of how fast to keep your foot on the gas, whether it's all the way down to the floor or not. And then if you're going to have your foot on the brake or near the brake, how often you tap that brake and or not, right?
Yeah. Yeah.
Okay,
I tell docs, hey, when you give these drugs, who's the brakes? I don't know.
exactly.
No, you're the brakes. You pick up your phone in the middle of the night when the patients call you. You're the brakes. You're responsible
Exactly.
for that trigger. So the gas pedal here is the IL-2 and the interferon, which are kind of self-made in the body. And then the future where immunotherapy is going again is kind of reintroducing vaccines in the setting of release the brake and gas pedal so we can steer the immune system. So you're seeing a lot of things around like neoantigen vaccines,
Yeah. A
trying
lot of
to
mRNA.
yeah. And this is also why cancer vaccines didn't work during the 70s and 80s and most of the 90s, because we didn't know the breaks. We were just giving cancer
Yeah, it was
vaccines,
just full throttle.
inducing
No, it was all or nothing,
yeah,
right?
we were just inducing more tolerance. Just like if you go see an allergist, they give you little doses of the allergen to make you become tolerant. You just become
Yeah,
tolerant.
see if it works, and if it
Yeah.
does, okay, we can give you a little bit more, but we just don't wanna give you the full throttle dose all at once without any knowledge of how you're gonna react.
So now a lot of those old cancer vaccines are trying to look at them in different ways now in modern immunotherapy. So let me see if we
Makes
can next
sense.
more slide. So this is kind of how we look at the modern immune system of self versus non-self. So the immune system, it's very smart, but it has to figure things out on its own. So the immune system can easily differentiate a very foreign pathogen that is non-self. Like I use the analogy, you step on a thorn, your immune system knows, oh, that is a piece of wood. That ain't human. That ain't mammalian. That is nothing. That is easy to attack. Or a virus, relatively easy to attack. Or some other pathogen parasite, et cetera. Or somebody does an organ transplant that doesn't match your body's tissue type. Easy to attack. It's very obvious. But when tumors grow, tumors are 99.999999999% self. So it's hard for your immune system to recognize it as foreign. And so the default of the immune system is to use the CTLA4 to tolerate self. Just tolerate self. Self host, we are too valuable. Just tolerate everything. So most of our immune system, believe it or not, are Treg cells, which go around tolerating
Tregs.
self. They just tolerate ourselves. And people with autoimmune disease, their Tregs and their self-tolerance balance is a little off. And they have their own suffering because autoimmune disease is equally a chronic suffering disease, almost as bad as, if not equally as bad as cancer. Where in cancer, though, we need a little bit of autoimmune disease. So we tip that tolerance the other way. So we get a little bit of autoimmune disease to release the brakes, overcome self-tolerance, so-called break self-tolerance, and mount a robust immune response and hopefully give vitiligo, right? So that's a good, and this is just a reference. I'll zip through this. This just shows more at a diagram. This is the references for your audience. They can screenshot and look those up in the literature. Very good examples. We authored some of these good teaching articles. So just showing the early
Great
days. And this is
visuals.
awesome. Remember I told you, don't give the drugs to pregnant women?
Yeah.
So we joke with patients, or I like to joke with patients. I hope they laugh at my jokes. I
I
say,
love a good joke.
dear patient, tumors aren't that smart, right? They aren't that smart to invent their own proteins, right? So they just steal from places in biology. So if we look really closely at biology, we get these great clues, like in the pig and in the horse and other places where we can get clues and study embryology, which recapitulates ontology, which is kind of like over evolution. And tumors do also. People don't know this, but when a tumor is born as a cancer stem cell, it also goes through kind of its own way that it grows like a placenta, and it expresses the same markers like a placenta because it knows how to hide like a placenta from the host. And so here's a placenta. Guess what's chock full on its surface is plenty of PD. So there's so much PDL1 on placentas. We use this as our positive control when we're doing studies on PDL1 and PD1 in human research. So, and here's a joke. There's the tumor cell, right? The cancer cell that's it's expressed in PDL1. It didn't invent that. It turns on that gene because it's buried deep in its genome. And as it becomes more a cancer, cancers kind of go reverse in time to become more embryonic and express those old embryonic proteins, which are conserved all the way through evolution, way back to the worm. So, and then there's little round immune cells on the surface trying to attack that cancer cell, but they can't as long as that PDL1 and PD1 handshake is going on. So modern checkpoint drugs go in, they block that or release the brakes to unleash immune cells to proliferate. So I just wanted your audience to see that. So this is just a good
It's a
teachable
good visual, man.
so
Yeah.
this is like we did the first dose of pretty much all these drugs. It's our claim to fame. That's the one thing we do good is doing a lot of these early trials, figuring out the side effects. This is one of the drugs of
We
our
don't do
first
that here.
patients. Early, like among the first patients that got the drug, had melanoma, right? Like you, but not mucosa. It was in his flank or something. And this was his scans looking back from March to May. You could see the measurable tumors and how they had shrunk over time. And then, especially the ones we could palpate and feel. And some of them, as they were shrinking, we were able to, first he got vitiligo. So it's like a few weeks into the treatment, he got an itchy rash and he was getting immediate vitiligo. He was breaking self vitiligo. And then as he was getting vitiligo and as his tumor was shrinking, we did a biopsy of one of his tumors and you can see the hand-to-hand combat of the immune cells killing, which is really cool. So we just show this here. So this is, we call the immune front. This is how we say there's hand-to-hand combat of the immune system killing. Remember I said melanoma is easy to tell under the microscope? Where's your pigment? So the pigmented melanoma is way on the right-hand side, right? So on that right-hand side, the pigmented melanoma, all those little blue round dots, those are the lymphocytes that are going from left to right, attacking the tumor. And the tumor that they had already killed is what's left over in the left background. That's the debris
I was
of
gonna say, you
leftover
can almost, like,
tumor.
literally see it, like, the wave taking over, you know?
Literally, it's like a day by day hand-to-hand combat, cell-to-cell killing. In the kinetics of this scientific community, they are working out the kinetics of this almost down to the day and to the hour. And the molecular machinery around how some tumors even try to evade this, or how some tumors become stemness and dormant, and they just become like a dormant little seed and can hide. Even though 99% of the tumor may regress, dormant seed may hide. We're sort of learning more about this, and we did a lot of biopsies on these early days. And me as a clinic doc, a small clinic, we even got published in Nature with this. It's a good article, it
Yeah.
shows great pictures. So it's just to show you that there is, it's hardcore science. It's very real. You got to see it to believe it. Immunology is not a black box.
No.
It's very real. This is just another case of a different checkpoint drug. This is a lung cancer patient, his tumor disappearing. This is a great picture of the two early discoverers, scientists who discovered the checkpoints, the CTLA4 and the PD1, the Japanese physician Tasuki Honjo and James Allison, the checkpoint, the CTLA4. And this got on the front page of Nature and Science. We were published in the Nature article, and for our biopsies of the immune front. And then Dr. James Allison won the Nobel Prize. The two of them won the Nobel Prize together. And our clinic actually went to celebrate him winning the Nobel Prize at the Checkpoints band when they played at the SITC conference.
Yeah.
So that was kind of cool. So next is we'll pause here and wind down. But then our next talk will talk about how we are trying to overcome where the checkpoint drugs don't do a complete cell kill. So that'll be for the next slide. That's a good review of what we do.
Man, that was mega cool. For those that don't have necessarily a background, like this is a huge visual to just kind of get like I don't know, let's just say a 30-year summary of where we're at now. And we didn't even get through all of that. We just got to a certain section of it. So next episode, we'll pick up where we left off. But it all has to start somewhere. And I always say a little bit of something is better than a little bit of nothing, but it all starts somewhere. And these are your cells. I mean,
these are your immune
John,
cells.
I mean,
That's these checkpoint drugs. They don't do the killing. They teach your immune system how to kill your own cancer, which is a big paradigm shift. So, and then next time we'll talk about how we're trying to grow your own immune cells so we can mount a bigger immune response by growing them up in a big, factory.
Instead of hiring somebody else's army, you're getting your own army from you. So you can kind of, we'll dig
In a way,
deeper
it's
into
the most
that.
naturopathic, really, it's really leveraging your own immune system, which I think is why it's so transformative in the future. Those would
That's
be good things to follow
so
up
important.
on future talks. And yeah, Chris, as always, I love the chance to speak to your audience and do some educational teaching about the historical research that contributed to the development, standing on the shoulders of giants to bring you the TIL and the checkpoint drugs and the next wave of cellular immunotherapy.
I got to tell you, John, this is definitely one of my more favorite TIL talks, to give some visuals to where it's not just been audio, but to watch and visualize it, and to be able to explain it in simplistic terms. And thank you for coming on and showing this. We continue and pick up on the next one in January. Again, John, thank you for coming on TIL Talks. Thank you for Carolina Biooncology Institute, Biocytics, and everybody, and always peace. All right, that concludes today's episode of TIL Talks with Chris White, as we talk TILs, tumor-infiltrating lymphocytes, and all advanced cell and gene therapies. As a reminder, I am not a medical professional or medical expert. I am only a professional and expert on my story and my experience. If you are needing or seeking medical advice or assistance, please reach out to your care team. I'd like to thank my guest today for joining me on TIL Talks. To our listeners who have not yet done so already, please follow, comment, subscribe, share, and smash that like button. For more information about TIL Talks and related resources, please visit www.tiltalks.com. I'm Chris White. As always, sending love to everybody. Peace.